Hello, my name is Chris Bunker. I'm the Senior Director of Business Development at Advanced Cell Diagnostics, which is a brand of Bio-Techne Corporation. Today, I'm going to speak about our RNAscope in situ hybridization assay technology in relation to expanding clinical biomarker development options. RNAscope is a clinically validated quantitative in situ RNA biomarker platform that we've used in the past and currently for the development of many clinical biomarker assays and potential companion diagnostics. As an organization, we focus primarily on developing and designing novel probes for detection of essentially any transcripts in fixed tissues. There are nearly 30,000 probes in use currently, and these have been used extensively in a very wide range of research applications. The primary features of RNAscope in situ hybridization are, first, that it is intended for and enables analysis of expression in formalin-fixed paraffin-bedded tissues. These can be archival tissues stored for up 10 years or more, or freshly collected from clinical trials or any sort of animal study. In all cases, every probe enables single molecule detection of a transcript. And that is to say, we can visualize individual molecules as dots, quantify those on a single cell basis in intact tissue. Moreover, it is extremely specific. Much like any other nucleic acid-based method, the specificity can be predicted and calculated prior to actually making the probes. The assay itself is very similar to either IHC or immunofluorescence. It's slide-based. It could be run on plates as well, but primarily slide-based, starting with tissues or cells on a slide, and then hybridizing first with oligonucleotides specific for the intended transcript or transcripts, and then amplifying signal through a series of hybridization events that will eventually localize thousands of molecules of label to a single RNA transcript. And this signal can then be visualized and quantified using image analysis software. These assays are all kit-based. Any probe can be used with either chromogenic detection reagents or fluorescent detection reagents. These assays can be run fully automated on Leica or Ventana autostainers or the benchtop hybridization oven that we sell. ACD is primarily a probe company. We have nearly 30,000 probes in our catalog, and the strength of the technology is the fact that we can design probes where we have essentially certainty about their specificity before we even make them. That's true of every single probe in our catalog. All the probes are built to the same specification, knowing that they're all going to be used under exactly the same assay conditions. So that when we make a probe and design the oligos that comprise the probe, we can determine the specificity based on calculated melting temperature for every single oligo and every single probe. Now I'm showing you schematically here a number of things. First, the probes themselves consist of oligonucleotides, and we speak about those oligonucleotides functioning in pairs because signal amplification is dependent on paired oligonucleotide hybridization to a target sequence and then paired oligonucleotide hybridization to amplification modules. And that dependency on two oligos hybridizing right next to each other to amplify signal is how we build in specificity while amplifying signal to nearly 1,000-fold per oligonucleotide pair. As you can see on the right, a typical probe, a typical RNAscope probe, will consist of approximately 20 pairs of oligos tiled along a region typically about 1 kb. Now here again, there are features of this probe design which make it particularly useful for analysis of RNA in fixed tissue, formalin-fixed paraffin-bedded tissues. The fact that each one of these oligo pairs is binding to a very short sequence, about 50 nucleotides, and there is a very modular structure, these pairs can be moved around and positioned to ensure single target specificity. In addition, since they're only binding to 50 nucleotides, the probability of that binding site being accessible to probe hybridization is much higher than if you were to use or if you needed a longer sequence, say for PCR-based amplification. In addition, we use many more oligonucleotides in a probe than are actually needed to detect that transcript. We only need about 3 pairs of oligos binding to a particular transcript to generate detectable signal. So by using approximately 20 or so, we have a very high level of redundancy, which translates into a very high level of molecular efficiency for detection. So RNAscope is our primary assay we speak about. And BaseScope is another assay, very similar in terms of the principles of detection and signal amplification. And I'll speak about the cases or the applications for one or the other. So by default, we would generate typically an RNAscope probe. These probes, again, work for the vast majority of target sequences we might want to detect. They're used in research. They're actually used in vitro diagnostics as well. And we have quite a bit of flexibility in what we can do with RNAscope assays. We can do single color or two color chromogenic assays by using one or two probes combined. We can do 3 or 4-plex fluorescent assays. And we have a variation assay called high-plex, where we can actually do up to 12-plex fluorescent detection. That's limited to frozen tissues for its use, whereas all the other assays are intended for formalin-fixed tissues. Now, BaseScope is applied in situations where RNAscope cannot generate either a specific signal or enough signal given either sequence length or homology. So with BaseScope, we need just a single pair of oligos to generate detectable signal, and that's in part because the amplification module is much larger. We get about 5,000-fold signal amplification with a pair of oligos with a BaseScope assay, whereas with RNAscope, we get about 1,000-fold signal amplification with each pair of oligos. So BaseScope also has slightly different probe chemistry, more like locked nucleic acids, which enable us to differentiate much more highly homologous sequences to the point that BaseScope can actually be used to differentiate a point mutated sequence from a wild type sequence. It's also very applicable, and we use it extensively in gene therapy research, where oftentimes the desire is to detect a human transgene in the background of either mouse or non-human primate ortholog sequences in that particular tissue type. So there are specific cases where BaseScope are used and other cases where RNAscope is perfectly suitable. Now, again, just to kind of walk through schematically how signal is amplified with RNAscope and BaseScope assays, again, it depends on paired oligonucleotides hybridizing to a target sequence, and then those pairs also hybridizing to a preamplification module, which then hybridizes to amplifiers, and then labeled oligonucleotides, either labeled with HRP or alkylating phosphatase or fluorophores. And as I mentioned, this amplification module gives us 1,000-fold signal generation per pair with RNAscope, and with BaseScope, it gives us 5,000-fold signal amplification with a single pair of oligos. And this amplification only occurs when pairs bind, which results in a very clean assay with essentially no background under typical assay conditions. Now, we mentioned single molecule detection is what we focus on and what's possible with the RNAscope assay. This is illustrated in an experiment that was done in the first publication to demonstrate the RNAscope assay. Here, the experiment was to take two helix cell cultures. In one case, do an RNAscope assay with a HER2 probe, which shows up in green fluorescence. In another plate of helix cells, the RNA was extracted and measured in a solution-based assay where a standard curve of in vitro transcribed message could be used to determine the average copy number of HER2 transcript per cell in that extracted RNA. And what you see here is that you get very similar numbers, whether you count dots in the left in a field of view, you get about 14 dots per cell, whereas on the right-hand side, if you measure in solution with a standard curve, you get about 17 copies per cell on average. So the primary point here is that when we see a dot, it corresponds to a molecule of transcript in this case. Now, to further illustrate the point that a signal generated with RNAscope as a dot represents a molecule of nucleic acid, if this same assay is run under conditions which more extensively permeabilize the cells and we remove RNA and expose the DNA, chromatin DNA, the same assay will generate 2 dots for the 2 genomic loci in HeLa cells. You can see that in the left-hand panel. The right-hand panel is a cell line which is amplified for the HER2 locus, and you see multiple dots. So here again, we're simply making the point that a dot corresponds to a molecule of nucleic acid, in this case DNA. In the previous case, we were showing RNA detection. Now, to put this in the context which is a little bit closer to the focus of this presentation, which is on clinical biomarkers and diagnostics, here further data comparing RNAscope and other methods for HER2 analysis. Now, what's shown here is an example of a number of cell pellets known to express or not express HER2. What's shown on the left-hand side are data with RNAscope assays, control probe assays on the left and the middle, and then a HER2 assay for RNAscope shown on the right-hand side of the panel of 3. And what you can see are progressively increasing signals with the HER2 RNAscope assay from very low to very high. And adjacent to that is a panel of the same cells tested for HER2 by IHC. And what you see, it's low magnification here, but there's no detectable HER2 in the top panel, whereas you can see increasing amounts of DAB stain in the lower panels with that antibody. Now, that same antibody will in fact detect HER2 protein in the first sample, core number 1, when it's used for Western blotting, which speaks to the fact that Western blot is oftentimes more sensitive than IHC. And in this case, the RNAscope assay is showing detection in a cell pellet, a visual assay using in situ hybridization, which correlates with what's shown in the assay from extracted proteins in a Western blot. And the IHC assay in this case is a less sensitive readout. Now, we don't necessarily plan to sort of reinvent the wheel for HER2 and develop a diagnostic for this, given the fact that there are well-established diagnostic tests for HER2. However, it is worth considering for some companies that are developing new HER2 therapeutics that are either based on antibody-drug conjugates or some cell therapies. Now, these modalities may be more potent and may be effective on tumors where expression is quite low. Expression may be sufficient to engage a potent therapeutic like a CAR T cell, but that molecule of protein and proteins there may be undetectable by IHC, and a more sensitive assay like RNAscope may be effective for identifying and diagnosing those patients. This here is some further data that relates to the quantitative nature of RNAscope in situ hybridization. This is work from the late Ray Tubbs at the Cleveland Clinic from a few years ago, and work that he did was to try to establish a way in which to resolve equivocal cases of HER2 status in breast cancer. Now, established methodologies of FISH and IHC in some cases are equivocal in that they can't determine whether a patient is truly positive or negative. And so what the lab of Ray Tubbs did was use the RNAscope assay to define a molecular threshold based on a training set that they could call HER2 positive. So it's a molecularly defined threshold that's based on the percentage of cells that express HER2 transcript at a specific level. And the data that they reported was on the one hand showing a set of patient biopsies that were equivocal by IHC, which is to say IHC could not determine whether they were truly positive or negative. They were negative by FISH, and RNAscope correlated with that FISH outcome and also confirmed negative. In the cases where FISH was positive, again, IHC is equivocal. What's shown here is good correlation between RNAscope and FISH for determining positivity in terms of HER2 status. Now, the final column, the final data set are the set of patients that were equivocal both by IHC and FISH. Neither one of those assays could resolve them as being positive or negative. And what the lab of Ray Tubbs showed was that again, a molecularly defined threshold of positivity could resolve those cases into either truly positive or truly negative. Now, we of course are biased in our perspective that RNAscope is a very useful, potentially more useful methodology for clinical biomarkers and companion diagnostics, but I think it's fair to say that there are characteristics of the assay that do stand out against other more well-established methodologies. The morphological context of RNAscope is comparable to IHC, being able to identify specific cell populations within a tumor biopsy that are positive or negative or dual positive for various markers is a benefit of a visual histological assay like RNAscope or IHC. The sensitivity and specificity will oftentimes be much greater than IHC, more comparable to a molecular assay. And the advantages over molecular assays, on the one hand, is that in the first place, it can be run on routine formalin-fixed paraffin-bedded tissues, which is typically not the case for most molecular extraction assays. And in terms of tissue requirements, only a few 5-micron sections of tissue are needed for an RNAscope assay like IHC, whereas oftentimes a much larger amount of tissue is required for molecular extraction-based assays, and that amount of tissue is often not available if the sample type being collected is, say, a needle biopsy. For the development of RNAscope-based diagnostic tests, Advanced Cell Diagnostics has a partnership with Leica Biosystems. With Leica, we are in the process of developing a number of companion diagnostics. These are still fairly early stage, but these are in development, and if the corresponding drugs are approved as therapeutics, these would lead to our first companion diagnostics. So again, we have a very well-established relationship with Leica, and this is our channel through which diagnostic development would occur with the RNAscope technology. The benefit of working with Leica is the fact that their automated histology systems, the Bond RX and Bond 3, are very similar, essentially identical instruments. The RX is what we and many other research laboratories use for research grade tests and assays with RNAscope. The Bond 3 is an essentially identical instrument. It is FDA approved, system development is under design control, and assays can be fully validated and registered with the FDA based on being performed on that instrument. At Advanced Cell Diagnostics, in our pharma assay service laboratory, we develop biomarker assays and assay protocols in a research environment. We are not a CLIA lab. We will be CGLP compliant in mid-2020, but we are not suitable for doing prospective testing with the RNAscope assay. For that purpose, we have a number of well-established relationships with CLIA labs, which we've certified, trained, and worked closely with to transfer assays developed in-house at Advanced Cell Diagnostics into these laboratories for use in prospective testing of patient biopsies. In the next few slides, I will show a number of case studies that illustrate where RNAscope has proven to be a more effective means of detecting a biomarker that is predictive or appears to be predictive of patient response to a cancer therapeutic. The first program we worked on was with Merrimack Pharmaceuticals and their anti-ERB3 antibody therapeutic called MM121. They had determined that the ligand growth factor of ERB3 was the biomarker that was predicting or at least correlated with patient response to treatment with an anti-ERB3 antibody in combination with standard of care chemotherapy. This was based on phase 1 studies where they saw the correlation between the expression as shown here, actually very low level expression in some cases, in the tumor itself that was predictive of a tumor which was driven by this growth factor signaling. What I show here actually is 3 assays, 2 important control assays that are in part of every RNAscope assay, and that is using 2 controls, a low copy housekeeping gene and a negative control bacterial gene to eliminate the possibility of false positive or false negative results in the assay by first verifying the quality of the RNA and the quality of the sample prior to testing the biomarker itself. So this assay was used in a phase 1 study where they showed very good correlation, significant correlation between RNAscope heregulin signal and response to treatment. And that was their basis for partnering with Leica Biosystems to have this assay developed as a companion diagnostic for use in a prospective test in a phase 2 clinical trial. So just to show the data, again, this is from their phase 1 study, retrospective data comparing patients treated with patients treated with MM121 in combination with standard of care, and looking at those patients, subpopulation of patients shown in blue, which had high levels of heregulin expression. So in breast cancer, ovarian cancer, and lung cancer, in each case, where heregulin expression was high, there was a benefit to combining MM121 treatment with standard of care chemotherapy. This is simply a screenshot from the ASCO 2019 poster presented by Merrimack and their academic collaborators reporting on the results of the phase 2 clinical trial study for MM121. And I use this only to emphasize and illustrate the performance characteristics of an RNA in situ hybridization assay applied to clinical biopsies. In this case, the performance was very highly reproducible and robust on a set of patients, nearly 500 patients coming from 87 different clinical sites on 3 continents. Another example where RNAscope has proven to be the most effective method for biomarker assessment is for the receptor tyrosine kinases FGFR 1, 2, and 3, for which Bayer has a small molecule tyrosine kinase inhibitor. RNAscope was shown to be superior both in terms of specificity and sensitivity against IHC and nanostring assays. Originally, it was reported in 2014 that RNAscope FGFR assay was a better predictor of FGFR tyrosine kinase inhibitor sensitivity as compared to gene amplification, and it was also shown to be more specific than was IHC. Based on these results, Bayer used the FGFR mRNA assay as a basis for enrollment of patients in a phase 1 clinical trial. Positive results in that phase 1 clinical trial led them to partner with Leica Biosystems to have an FGFR companion diagnostic developed based on the RNAscope technology. This assay was then used for patient selection in a pivotal phase 2, phase 3 clinical trial run with the RNAscope assay developed as a kitted test by Leica Biosystems. Just as an anecdote, shown here is an example of a patient which was highly responsive to the Bayer therapeutic tyrosine kinase inhibitor in an instance where there was no indication of FGFR amplification. However, the patient did show a high level of expression of FGFR in the tumor biopsy. Just to further illustrate the point I made previously, this is another snapshot of an ASCO poster describing the use of measurement of FGFR RNA expression as the basis for patient selection in their phase 1 trial. This trial involved 860 patients on which they ran FGFR 1, 2, and 3 tests. Again, illustrating the performance characteristics and high level of reproducibility and robustness of a clinical RNA in situ hybridization assay. To provide context for the next biomarker assay case study that I'll speak about, I will reference a press release from Ventana from 2013 describing their intention to develop a companion diagnostic test for Insight for their small molecule IDO1 inhibitor. And this is a context in which to reemphasize the fact that oftentimes RNAscope will provide a more sensitive assay than IHC. In addition, it provides a much faster timeline for development and verification of assay performance for use in a clinical context. So this assay, the IDO1 IHC assay being developed by Ventana, was begun in 2013. In 2018, Insight presented a poster at ASCO SITC describing the sensitivity and specificity of the RNAscope platform for detection of IDO1 in tumor biopsies. And this was after a very short period of development work that we did with them, in which we demonstrated to their satisfaction that RNAscope was about 10 times more sensitive than the IHC assay that they had spent many years developing with Ventana. Now, this is certainly no statement against Ventana or the performance of their antibody development. It just is a statement about the ability to develop an assay very quickly with RNAscope, which in many cases may be more sensitive and or more specific than IHC. This is a little bit more detail on the assay and its performance in a cell culture system, HeLa cells, which can be induced to express IDO1 with interferon gamma treatment. What's shown here are comparison data for IDO1 measurement by RNAscope, by IHC, and by Western blot. And the plot on the right-hand side illustrates the fact that there's a near perfect correlation between IDO1 RNA as measured by RNAscope and IDO1 protein as measured by Western blot. However, the assay for IDO1 as measured by IHC is about 10 times less sensitive based on the fact that it took a 10 times higher dose of interferon gamma to induce the same level of detectable expression by IHC. And it was for this reason that Insight switched from the IHC assay, which they'd spent a lot of time and money on developing, to RNAscope for use in a very large number of clinical studies in many phase 3 programs. This, again, is data to emphasize the performance of our assay across a very large number of patient biopsies coming from multiple different studies, multiple different tumor types, uniform performance of the assay across all these different conditions. And despite the unfortunate fact that the IDO1 compound in the end did not show any efficacy or improvement in terms of patient condition or response to treatment, the assay itself performed extremely well across a very wide range of patient biopsies in a clinical trial. The case study of IDO1 and the performance of RNAscope in measurement of IDO1 RNA is a very good case study from a technical standpoint. It addresses a question that we get routinely about how RNA and protein correlate. Now, it showed very clearly that RNA and protein correlated very well for IDO1 when you had equally sensitive assays, namely Western blot for protein and RNAscope for RNA. However, nowhere in that study was there an assessment of how expression correlated with response to treatment. And in the end, there did not appear to be any correlation between those. In the case of the first study I showed for Heregulen, for FGFR inhibitors with Bayer, and here again with Leap Therapeutics in data that they presented at SITC in 2019, they observed a very good correlation between DKK1 expression and response to treatment. And they are actually going to be using this as a patient selection test in an upcoming clinical trial where patients will be enrolled based on having a certain threshold. The last example I'll provide is, I think, our most compelling case for the value of RNAscope and RNA in situ hybridization as a diagnostic test. And as the title here implies, this is from a review article from key opinion leaders in the field of head and neck cancer diagnosis for HPV. The same conclusion has been reached by the College of American Pathologists, which is that the measurement of HPV oncogene E6/E7 transcript is the most specific and sensitive methodology for identification of head and neck cancers where HPV is the driver of that cancer phenotype. This is in comparison to multiple other tests which have been in use for quite some time. The three methodologies that existed prior to RNAscope came into being were DNA SISH, P16 IHC, and qPCR. Here we're showing nearly perfect concordance with qPCR for measurement of HPV transcript by PCR or by RNAscope. We would argue that the benefit and value of the RNAscope assay over qPCR is the ease with which samples can be prepared and how FFPE samples are much more routinely collected in a clinical or diagnostic setting as compared to the sample type needed for qPCR. In comparison to P16 IHC, which is also very widely used, the data from recent publications and many previous publications points to the fact that P16 is not always a reliable surrogate biomarker for HPV status. As illustrated here, the primary point here is that there can be cases of P16 positivity where the cancer is not in fact driven by HPV, and that's in blue, you can see the blue line there, which tracks with the black line. P16 negative as well as P16 positive HPV negative had the same survival outcome compared to truly HPV driven and the samples which are both P16 positive and HPV positive. The conclusion and point here is that simply testing HPV positivity based on RNA signal, RNA measurement will address both of these data points and provide the most clear, specific, and sensitive methodology for HPV diagnosis. So this test is currently not FDA approved, but being used in over 100 reference labs in the US and now being adopted in Europe. Over 40,000 tests per year are run using our RNAscope HPV test for routine diagnosis and determining the status of HPV as to whether it's driven by, the status of head and neck cancer as to whether it's driven by HPV or not. this exact same test has equal use in the context of cancer vaccines targeting HPV and TCR T-cell programs targeting HPV. The same sort of specificity and sensitivity which is demonstrated in a prognostic test would be applicable and of value in targeted therapeutic approaches and cancer vaccine approaches just as well. Here I want to remind you that we have two assay technologies, RNAscope and BaseScope in situ hybridization assays. I've spoken and shown you data for biomarker assays based on RNAscope. That is our primary assay. That's the assay which is applicable and useful in the vast majority of cases where an expression biomarker is needed. The assays that I've shown you are just a few among tens of thousands of probes in our catalog. The fact that all those probes work under identical conditions, have essentially identical affinities and single target specificity mean that any one of them can be evaluated very, very quickly as to their performance as a clinical biomarker. Whether or not you have other orthogonal methods you may be considering, whether they be IHC or PCR or other methods, it is a very simple and time efficient process to evaluate the performance of RNAscope compared to any other assay technology. I should emphasize also that while I've shown you just single marker readouts in the previous slides for biomarker assays, this assay and any other assay which involves one or more targets can be evaluated in either chromogenic readouts with 2 colors or 3- and 4-plex assays with fluorescence. The BaseScope assay, which I'll show you a few examples of as well, has specific characteristics that make it most suitable for detecting targets which may not be as suitable for RNAscope analysis. These include point mutations such as KRAS mutations, sequences that are very highly homologous such as the MAGE family, which are cancer vaccine and TCR T-cell targets, and mutations like those in EGFRB3 or the MET tyrosine kinase. I'm just going to show you a few snapshots of data to illustrate examples of duplexed and multiplexed assays using RNAscope, and then I'll show you some BaseScope examples as well. So this is simply an assay looking at ADORA2A and its and CD73 in tumor tissues. This is 2-color chromogenic assays. Just an example, we can combine any probes in duplex assays like this. We can just as well combine additional probes. Here we're showing 3-color to look at 2 immuno-oncology targets, IDO1 and PD-L1 in combination with CD8 immunofluorescence. We could just as well add in a 3rd marker or look at 4 markers simultaneously with 4-color fluorescent in situ hybridization. This is true for the markers shown here, and any other probe in our catalog can be combined in a similar assay without any real assay development to be done. The BaseScope in situ hybridization assay is the other platform that we have available for biomarker development. This has very specific applications in cases where RNAscope is not applicable, either because the target region is too small, too short, or because of a very high degree of homology between the intended target and non-target sequences. This assay, as I illustrated previously, can use as few as a single pair of oligos targeting as short as a 50 nucleotide region. This allows us to detect targets such as exon junctions or fusion junctions, internal deletions. We can also differentiate very highly homologous sequences such as the MAGE family or NY-ESO, and we can even detect point mutations such as KRAS. I'll illustrate that in the next couple of slides. Here is a BaseScope assay showing detection of KRAS mutant and KRAS wild type in different regions of colorectal cancer tissue biopsies. The red region is identified as being positive for the KRAS mutation, and the yellow regions are positive for KRAS wild type. Now, in most cases, KRAS and other mutation status is determined most often by NGS. That provides you with an overall view of presence or absence of a mutation. The potential benefit of an in situ hybridization assay, such as shown here, is to look at the clonality or local distribution of a mutation within a tumor. So here is a very high-level view at a higher resolution. We're showing here similar sorts of signal as shown in previous assays a dot-like pattern corresponding to detection of KRAS transcripts, and in this case, showing that assay in combination with cytokeratin immunohistochemistry stain to identify tumor regions. So there's actually a lot more data that goes into the verification and validation of mutation specificity. I'm not going to show that here, but we have data verifying the specificity of about 6 different KRAS mutations, as well as EGFR mutations, PI3 kinase mutations, and others, all of which have the same sort of single nucleic acid specificity and performance in FFPE tissues. Another application of BaseScope is in detecting internal deletions or junction sequences that result from either fusion transcripts or splice variants. Shown here is a schematic of probes used to look at MET tyrosine kinase exon 14 deletion. 3 probes are shown here schematically, and I'll show you data on the next slide. One is a probe common to both the MET delta 14 and the other sequence being MET wild type. That the 12-13 junction is common to both. The 13-15 junction is a mutation-specific junction created following deletion of exon 14. And the 14-15 junction is present only in the wild type where the exon 14 is still present. This is data on cell lines known to harbor either the mutation in the top, H596, or the bottom, A549. The common probe 12-13 is showing signal in both. The 13-15 probe is specific for the mutation, showing signal only in the top panel. And the 14-15 junction is specific for wild type. And then just finally, just snapshot data, not showing the full validation data package that would go into any one of these assays, but for targets like NY-ESO, MAGE-A1, MAGE-A3, and other members of the MAGE-A family, these are all targets for which there are other sequences that are very highly homologous, which we cannot differentiate with RNAscope. Also very difficult to differentiate with antibodies, and oftentimes difficult to differentiate with PCR as well. So these are, again, simply snapshots, not showing the full picture for validation, but just illustrating the fact that we can detect these important targets for TCR T-cell as well as cancer vaccine development. So the assays I've shown you are all based on kit products, off-the-shelf products, which is the majority of ACD's business. Also an important part of a business is our pharma assay services, where a lot of these biomarker assays are developed. It is a research setting. We will be CGLP compliant in mid 2020. We are not CLIA labs, so assays that we develop for use in clinical patient testing will most oftentimes be transferred to a CLIA lab. But we have the capability to develop, we probably have the best capability to develop any biomarker assay for use on either Leica or Montana instrumentation for then transfer to a CLIA lab for prospective use. And so in summary, the assays that I've shown you today are intended to illustrate examples of RNAscope in use as a clinical biomarker and potential companion diagnostic. We can develop assays very quickly, as I've said. It's a very short period of time in which to design a probe and put it to use and validate its performance either on archival tissues or freshly collected clinical biopsies. We focus mostly on single marker readouts for biomarker assays. However, it is a very straightforward procedure to multiplex assays to look at multiple targets simultaneously or looking at targets in combination with cell markers. I didn't focus on it too much here, but these assays can be quantified. Oftentimes, biomarker assays will be read out by a pathologist as an H-score. However, we can use image analysis software such as that from Indica Labs, their Halo software, to provide and perform quantitative analysis on 1, 2, 3, or even 4 biomarkers. So thank you very much for your time and attention, and I hope this will be of interest, and I look forward to speaking with you in the future. Thank you.